Thank you, Alfonso. Can everybody hear me okay? Good. All right, so find the clicker. Here it is. Got it. So I have to start with a confession, like every good fallen away Catholic. And that is, perhaps like many of you here today, I'm not a biologist. I'm not even a computational biologist. I'm an elementary particle physicist who somehow, by sheer accident, got swept into David Botstein's microwave vortex in the late 90s. And here I am today trying to understand something about biology still. Now, as a physicist coming into this field, I had a very naive perspective on biology, which I hope I share with some of you, which is shown here that we'd have some cells and biologists would do some experiments. I don't need to really worry about too much, but some nice data would come out. We get some parts lists, some parameters. You'd put that into some quantitative model the way you do in physics. You make a prediction, you compare that to your cells again, you keep iterating this until you have this beautiful theory, like a standard model of biology. Well, it's not clear that that's realistic biology or even very relevant, but nevertheless, I've presented this naive perspective. But in particular today, I'm with a group at Stanford that tries to do systems biology. It's the Shapiro-McAdams lab, and we study a little bacterium, and we're trying to understand it very completely and very precisely so we can sort of fit into this paradigm. But I'd like to talk to you today about my experiences as a quantitative scientist trying to rummage around inside the black box and see how I can contribute and what I can do. So we're going to talk about today. So we study this little bug called Caulobacter crescentus, and it's a gram-negative alpha proteobacterium. It's aquatic. It lives in freshwater lakes and streams. It's an obligate aerobe, which means it needs oxygen to survive. It doesn't have any anaerobic pathways. It's small physically and genetically. It's only got about 4,000 genes, about 2,000 of which are expressed at any given time. And it's got a couple of curious properties, which makes it an interesting model system. As you can see, it divides asymmetrically. What's up with that? I mean, it's kind of like a stem cell of bacteria or something. And it also is easily synchronizable. So you can get all these bacteria going through the cell cycle in exactly the same phase. And the cell cycles are very tractable, about 120 minutes, which is nice. So remember, these guys are small. Right? There's a mammalian helicell nucleus, and yet, you can pack thousands of these things inside a nucleus. So all the work we do, especially the fluorescence processing about today, has to be done with 100x oil immersion microscopy, which is a pain if you've ever done any microscopy. So there's a problem here. We want to understand this bug completely. We want a complete circuit. Now, they've done a lot of in metabolic networks. Good for things. Bugs do more than just metabolize. They swim, they divide symmetrically in this case, they separate their chromosomes, all kinds of things. This bug is useful for because of the synchronizability. Push the button. Yeah. So there's a problem with PleC wiring diagram easily because there's a problem with biological dark matter. There's genetics. It's just hard to find and hard to figure out. There's either no discernible phenotype. You have a very short coding region, so it's hard to hit it in a genetic screen. There may be essentials involved in redundant pathways. And unfortunately, some of the proteins involved in the subtleties of cell cycle regulation fall into these categories, and we just haven't been able to hit them. So here's a picture of this asymmetric cell division. The curious thing is that let's start where the red dot appears on the chart, okay? We call that a mother cell. It has a sticky holdfast. It's sort of bound to a substrate. It grows. It is replication competent, which means it can divide its chromosome. Okay? It's locked into only making one chromosome per cell division as opposed to a lot of other bacteria. It can grow, and it can divide, and it produces a flagella at the far end. And at the time of cytoplasmic compartmentalization, one half swims away, the daughter cell, lives a life aquatic. It's chemotactic for a while. Whereas the other side, the mother cell, immediately becomes replication competent again and can grow and divide. The daughter cell swims around for a while, presumably trying to find more suitable environments, and then through another switching mechanism, which we don't fully understand, it drops its flagella, pulls in its pili, grows a sticky holdfast in a stalk, and then only then replicate its chromosome and finish that part of the cell cycle. So there's some curious switching design going on here. Now, one of the switching mechanisms we think we understand, and our understanding was solidified by some work by the Jacobs-Wagner group at Yale University. And there's this idea here. You have two signaling histone kinases on two halves of the dividing cell. We'll just call them the red and the green. You don't really care about the names. And they're talking to each other through a response regulator, div-K. Doesn't really matter what the name is either. But it's taking this phosphorylation message back and forth between the two halves of the dividing cell. Now, you can clearly see that when that cytoplasm pinches off, you've closed the door, message can't get through. You get a lot more phosphate on one side on div-K and the dephosphorylated version on the other. Those induce two different genetic transcriptional programs in the cell, and that's how you get the switching mechanism. It's not so clear how you get the other switch, where you just have this free-swimming cell that sort of spontaneously decides to turn into a mother cell. Is it a countdown timer? Is it some slowly degrading protein? What is it? We don't really know yet. So here's sort of what the situation was up until a few years ago. We have this red and green protein, which are talking to each other through the response regulator. The abundance of that response regulator in the two halves of the cell either inhibits or allows this black box pathway, this signaling pathway. People have fleshed this out. I'm not going to go into detail because there's quite a few signaling molecules in there. That, in turn, either allows or disallows the phosphorylated version of a master regulator called CTRA to be present in the cell. And depending on the abundance of that master regulator, you either get chromosome replication, division, all that stuff, or you inhibit it. So what we're trying to do is we know there's more going on here than just this. All right. So what we decided to do was to use those two signaling molecules as reporters. And what we want to do is start from this sort of reporter set of fluorescently labeled reporters and some genome worth of other proteins, and at the end get some kind of ordered pathway for how this thing proceeds. That's the goal. The problem is we're dealing with subcellular microscopic phenotypes. We're dealing with the localization of subcellular proteins. So what we had to develop was a high throughput data acquisition and analysis program that would allow us to, in the white box, which I'll use the pointer now, right along here, scale up what are basically traditional microbiologist genetics steps to sort of the modern level. And then a new piece in the red box, which is the automated piece, looking at the protein levels in a subset way. Okay, so here's the reporter strain. We just labeled those two signaling molecules, plus we labeled this other structural molecule, which we know is important. Then you do Tn5 mutagenesis. Okay, so we perform conditional transpose on mutagenesis with an engineered Tn5 reporter, and it was specially engineered to have sequences on it that would allow us to do semi-random PCR and get all of the library of mutants sequenced at once. In fact, we also included Celexa primers on it, so we can in fact do high throughput sequencing on the entire library too. So we're going to get the whole genome. We're going to get the genome disruption of all of the genetic elements. Then you do this colony picking, which is pretty traditional by now, but a desktop colony picker, you don't have to go to the genome center. It's actually too cheap, but it fits right on your desktop. And it's nice and thousands of colonies. We developed this automated data acquisition analysis program. We have automated microscope and set of code which actually goes through and data extremely fast at because you have very small condenser and hard to maintain focus when you're moving over macroscopic. So it's sort of like subcellular fax. A fax machine, you use this reporter is present or not, but in the cell, the interpreter is changed. So it has no break on, except to say that in the end, we get a nice organized data structure that gets pipelined the offline analysis. So we don't just produce a bunch of TIFF files, which you have to figure out what later. We actually have a well-defined data structure which fits directly into the next piece, which is the analysis piece. And in this part, we do cell finding. We get the cell shape parameters with the cytoplasmic and the localized piece. It all goes into a summary data structure, and we completely leave the images behind that represent the cell. And you call this on a computer to make it go fast. It's nice because it's task parallel. It's just like old-fashioned processing. And there's another piece here which I usually don't mention when I'm talking to strict audiences, is to do a extensive cell simulation in Monte Carlo, because if you're this, as you know, you better make sure you're not automated it. So we need a way to our analysis is working properly and is systematically biased, and to compare different techniques to make sure we're using the best one. So we do a cell image simulation, which you just have to find a way to parameterize your cells. You know what this. So you parameterize the cell envelope, smear it with the optical resolution function of your optical system, point spread function, pixelate it according to the optics of your camera, get a simulated image, produce thousands of these images, and then run mini Monte Carlos, and then run that through your data analysis suite, the way you would in a physics experiment, to test it and make sure you've got the best possible software going. And the nice thing about this, if you plot a residual plot of your predicted, say, cell size or something versus the measured, once you put it through your automated analysis, is you can quickly see, well, you get the precision and accuracy, obviously, from the residual plot. But you can also see that you can do basically sub-pixel or sub-diffraction limited measurements of the position of these proteins. These proteins, remember, are just small clusters of molecules. And if you model that by essentially a point source of light, then you should have like a Gaussian distribution. So once you know that that's the model for the source of light, then you can say at sort of sub-diffraction limited resolution where those molecules are. All right, so at this point, you've got some numbers, which I sort of crudely represented here as a cell outline and some locations and intensities of fluorophores. But now you want to take, so we have quite a few numbers that come out from each cell. Now we want to say, put together the ones, or let's combine them in a way that gives us the best possible statistical power for our genetic screening, which is what we're actually trying to do. So in our case, we measure the localized and delocalized abundance, and some measures of the polarity or the localization of the signal. Okay, now what this is a flip chart of what's coming out. Let's go back to the screen now. Cell is an individual cell, and strips that's sort of separated by gaps are individual mutants, a separate genotype. And you can clearly chart running along there, and then boom, there's one that looks a little funny. This particular protein, in this case DivJ, is attached to it. That do things like WT for wild type. It's not really the homogenized reporter strength, so that we use those as control all the way through. So let's plot deep. This is all students, okay? So you get all in this space. Look at just the wild type controls from a somewhat tighter cluster. If you now say, we took a no holds barred approach, so we sequenced everything. We know where the disruption is and all of these things. All right, let's find some known target that we know we should hit, like that big pod J protein. So if you look at those, you see that they're distinctly separated from the wild type cluster. If you say now let's find one more TAC A, which we also know about, that's also well separated from the wild-type cluster. So we have some confidence that it's working at least in 3 dimensions. Okay, so now just like we've all done with microarray analysis, you can do genotype-phenotype clustering. Now, as opposed to microarray analysis, where usually the long axis is the reporter, the identity of the reporter molecule that you're using, or whatever gene, and the horizontal axis is the phenotype, could be the specimen or the time point in a time course. Here, I sort of turn that the other way. So the vertical axis is the disruption. Okay, it's the gene that we hit, and the horizontal axis across the top is the phenotype. So we use it's a huge cluster. Okay, we have possible phenotypes and lots of possible disruptions, but I've only shown here the most statistically significant and systematically robust of those. And again, here's the tricks, and you do that for all the reporter molecules, and we found these clusters. And the cool thing is that this cluster here pathway has factors which like this mechanism. The inferred work on here, and I haven't done anything as sophisticated as we heard in the last talk. This is just classic epistasis experiments that get you the ordering of the molecules or the proteins in the biochemical network. All right, so now this network looks something more like this, where I've sort of labeled the proteins with the colors that they were found in the clusters. So now we have a much more complete view of what this pathway is doing. I mentioned that it includes these signaling molecules here, transcription factors, and then this lytic transglycosylase as well. In addition, we found a new role for this CPE protein in PleC. So what we've been able to do is use microscopy to enable high throughput, high content live subcellular screening, and it's to find a partial phenotype that shows that this CPE protein is involved with the reestablishment of one of the green molecule, the PleC molecule. And that clustering phenotypes allows us to illuminate a complete functional network in a single experiment. And what we've done is go from the sort of unordered system you see on the left to the ordered one on the right. And we see this kind of picture a lot, where we have some node that a bunch of things feed in. But we won't go one step beyond that. And for us, it took classic epistasis experiments to get from that picture to the ordered picture. All right. So I want to keep that in mind. The readout's good because the readout's nice. The readout is exactly the same readout as for this screen. So you just do your epistasis and then put it through the automated system again and check if you get complementation. Okay, so I just told organizers that we should bring some stuff that was a little farther and say ask the question, can we want to get into our, we don't just want the part which happened, but we want some dynamic as well. So what we a operation system. So we use tetracycline repressor combinations encoded in the genomic DNA of the bacteria of known length. Okay, so we have a PCR technique by which we can generate different lengths of these teto dimers, insert them into the bacteria, and you can see how that looks here. There's a bunch of bacteria. Little spots is 20 or less. All right, this if I'm working in this field for all kinds of crazy schemes, you're much better off. So you can see where I'm molecules in them. plate with you're getting through your system. That here, a calibration curve is the actual. So let's test this. It's really good for cell cycle. Cells put on ice. They start to grow happily. And the cross you can run through the automated and get something like this. So on the vertical axis is the intensity of the protein I'm seeing, and on the horizontal axis, that's the time point. So I'm seeing the time development of this protein abundance. Now, you can do two things. You can use our calibration to change the vertical axis from raw signal to actual numbers of molecules. And you can also, because we have an automated analysis, you can get the phase of this and correct for it. And by using that, doing that phase correction and the calibration, you get something that looks more like this. Now, you can fit this with a sine curve, but a better model would be one that accounts for the sort of exponential decay of the degradation event early in the cell cycle, and then the sigmoidal increase in concentration as the cell cycle progresses. So that's what we did, and we feel that with a little more modeling, we'll be able to extract the kinetic parameters for this process directly from the data. So we hope to do something that's akin to in vivo biochemistry. So yeah, that's just a summary of the second piece. In vivo protein localized abundant measurements is possible and practical, and it's also, when you correct for cell cycle phase, we think we can extract kinetic parameters from this data. So I'd like to acknowledge my collaborators, Harley McAdams and Lucy Shapiro at the Department of Developmental Biology at Stanford, as well as Beat Christian, who is a really good molecular biologist, and every computationally oriented person should be so lucky as to work with someone like this. He's interesting, he's one of a set of twins who discovered cyclic di-GMP, which was a novel second messenger in bacteria, and they published that recently in Science magazine. And I also like to thank the NIGMS Quantitative Research Career Fellow Program, which allowed a physicist to go and dabble in biology. And finally, I'd like to close with a picture of our screening robots in action. The one on the right is the Swiss-built one, and the one on the left is the American one. You can see the automated. Thank you very much.
So how would you scale the system to culture the eukaryotic cells?
Eukaryotic cells? Well, it's funny you asked that, because we wrote a proposal to try to push this in stem cell biology. And I came back with lots of nasty comments, so I'm able to try again. But I mean, the idea being that there are symmetries. This is jaded asymmetric cells, the stem cells, asymmetries that are external to them, so they find them. They're also asymmetric. I think the thing is molecule. You're dealing with different microscopy, which I think is infected. Nevertheless, you have to did this well immersion, so but down transpose.